suppose a good first question, one that I ask everyone, is how did you first become interested in neuroscience specifically?
Yeah, it's a great question. I think I could trace it back to when I was 9 years old, when my school teacher, told me to watch a documentary about the brain on the TV, and this is maybe back in 1984 or so. Yeah. And it was quite fascinating because they were talking about how, the senses in the world are being processed, trying to answer questions like why do we dream, what happens in the brain when we dream, and somehow I think that sparked my interest. I could really trace it to that. Yeah,
I think that for a lot of people, neuroscience is unique in that it can tackle those sort of intangible things like dreams and sensations and stuff in a way that other life science can't really. It's quite a uniquely human science, I think.
It is, and because it somehow helps us understand our mind, the way we behave, that we sense the world, it might be easier to get into, or at least for me it was. It was a natural pull.
I think that makes sense. I think that also ties in quite well to some of your career around drugs and things, is that often they're a very human-centric kind of, it's related to experience and things like that, and so I think that's another facet of how neuroscience is useful, in that you can study perceptual things like that, which you can't really do in another sense. So what so obviously it's always a tricky question to ask someone to summarize their PhD, but how would you sum up your PhD? What did you study briefly?
So one of the things we did when I was a PhD student at the Free University of Amsterdam was use an animal model, try to find out how rats process cues, or these are signals which are related to heroin experiences as well as sucrose consumption, and to expose them to such cues using a particular behavioral task, and find out which brain areas are activated. And back then, when I did this project around 2004-2005, the hot topic was how do drugs activate the brain differently from natural rewards. So naturally, an extension of that was how do cues or signals associated with drugs versus natural rewards differentially activate the brain. And there were some interesting human studies based on that, MRI studies some years earlier, showing that cocaine-associated stimuli would activate the brain differently from, let's say, erotic stimuli. And it was kind of a cool study. I thought that they had a control for that, and they were saying, indeed, cues activate a particular brain area more in the prefrontal cortex. The thing about the animal studies was that we could use biochemical techniques and look at these activity-regulated genes called immediate early genes, which you've probably heard about in drugs brain behavior. So these are genes that are expressed pretty fast, within minutes, and they are activity marker, but they can also tell you what might be going on in neurons. For example, these are our particular signal transduction cascade that's being activated in neurons, are more genes going to be transcribed in the future, and so on. So we were using these immediate early genes, using a technique called quantitative PCR, or real-time PCR, to determine their levels of immediate early genes. And we basically found that with cues associated with heroin, there's more activity in the medial prefrontal cortex compared to cues associated with sucrose.
So when rats were exposed to sort of, you could say, memories or stimuli associated with a drug that activates a different part of their brain to memories associated with a sugar reward, what sort of implications does that have for what did that inform? Essentially, what's the main kind of takeaway?
The take-home message from that would be, because we measured this cue exposure and the brain activation some weeks after the rats last took sucrose or heroin, kind of shows you that these memories are very long-lasting, and they could be more robust with drugs of abuse. I mean, hence, if you look at someone who used drugs for many years, they're not going to forget about the memories or cues associated with the drug. So if you look at a smoker, they're never going to forget what that ashtray was about, or the feeling of lighting up their first cigarette in the morning. Yeah. So that would be the main message. Yeah.
I suppose that obviously has a big implication for people trying to quit smoking, for instance, or any drug of abuse. Because obviously if those memories are so sort of form in a different way and they're so long lasting, that can make it really hard to stop that reinforcing kind of behavior, I suppose.
And I would say it's in a way, for addiction, it's an unfortunate thing that they're so robust, so long lasting, and quote, hard to shake off. It's a different kind of memory from when we learn the biochemical pathways, like the Krebs cycle, where you study it, and maybe you'll know it for the test, but then some days later, you already forget about it. But so, I sometimes wish some of the memories, like the papers I've read, would stay long like that.
That's interesting. Actually, I wonder if there's a potential avenue of research there, in that if there was some way you could kind of trick the brain into forming memories that are as strong for non-drug associated rewards as they are for drug associated rewards. That's an interesting line of inquiry, isn't it? Maybe.
That would be good.
That would be handy.
Perhaps I think one way to do it is to make these, if you were to make something really salient by adding rewards to it, if someone were to say, I'll give you £1 million if you study the Krebs cycle and retain it for a long time.
That'd be nice.
that euphoria might make you remember that information better.
I think that would have made my undergrad a lot easier.
Right. It's a very costly way to do it, but it might work,
Definitely. So that was your PhD, and you're saying that's at the Free University in Amsterdam. PhDs are always everybody has a different story for their PhDs, but most people at university agree that they're quite difficult. What would you say you found? Was there an aspect of your PhD that you found particularly difficult, especially since you'd moved to Amsterdam from the United States? Was there anything in particular you found tricky from that aspect, or what would you say was difficult?
Not too tricky living in Amsterdam. It's quite a global city. It's a very open-minded city. It's a bit like being in Brighton. So in that sense, there's not too much to, quote, adjust to. I quite enjoyed my life there. Of course, the weather was not ideal sometimes. But I don't think there was too much to adjust to, except one cultural difference is that compared to Americans or even the British, the Dutch are more straightforward, honest, and direct in their discussions. So that was kind of a surprising thing in the beginning. They'll tell you straight to your face, I don't agree, but you shouldn't take any offense to it. That's them basically saying, this is where I stand. And we like to read each other that way in a more straightforward manner. But generally life was very good there. As for my PhD, I think the most difficult thing was for some years, my experimental findings were negative, which basically means the data you collect do not support your hypothesis. For example, if you say, I hypothesize that, the stimuli will activate these brain areas, and they just don't. Yes, that's a scientific method, but for publishing your findings, negative findings are generally difficult to publish, and that was quite demoralizing at times. So I really had to be persistent. There were moments, I'll be honest, where I felt like, I almost feel like quitting, because it's really tiring, because you do put in a lot of hours into it. Yeah, definitely.
So if you were to give a piece of advice to an aspiring PhD student, maybe it would be related to that persistence in the face of not failure, but not getting results that back up what you're looking for, like kind of negative results.
Be persistent in the face of adversity, fatigue. Of course, while working smart, as in, don't, if an experiment's not going to work, you better change your methods, for example, or you need to optimize things that you need to be smart about. But be very patient. Science just these experiments can take a long time. You have to learn how to optimize experimental conditions, work with various people, work as a team. It's a steep learning curve. In the end, it's kind of worth it, and because you also grow as a human being. Yeah. But yes, and did you also ask about advice?
Yeah. So if you were to give, if you had one piece of advice, or one back of the envelope, really pithy piece of advice, what do you think?
I would definitely say choose a mentor who is very supportive, i.e. is willing to kind of guide you through the process and then wants to truly develop you on an intellectual level, like stimulate you, make sure that your project is going to work out. Well, you can't, there's no 100% guarantee, but lowering the risk, but yet still coming up with something novel that everyone wants to learn about your findings. And that really is key, because once you have that, I think you're kind of set to the right path. Yeah. Yeah.
Well, I think that's good advice. Yeah. So, moving on past your PhD, after that you get to start as a postdoc. What was it like to go from that jump from kind of you're just a lowly, in air quotes, PhD student to then graduate and then get to start as a full-time researcher as a postdoc? What was that experience like? Was there anything you didn't expect so much, or?
Yeah. Well, one thing it was kind of an exciting phase of my scientific career. One, I felt like I had more. I was still doing science, doing experiments, but the project I was working on, it felt like at that time was going on the forefront, which was to, basically silence these neurons that were activated by, let's say, drug-associated stimuli, remove them to see, are they really involved in a particular drug-induced behavior? It was like science fiction, but I got to work on that project. So that was quite exciting. I had to work with at that time under two mentors who I had two projects going on in parallel. The amount and pace of work in my first 2 years felt like a lot. I had to adjust very, very fast. At some point I got used to it, but yeah, it did feel intense.
Yeah, I bet it was. It's not so easy when you've got two projects at once. That's a lot of work. So when you just start as a postdoc, and then obviously there's the process of going through that phase to having your own lab and your own group, and doing your own research. This is always a tricky question, because obviously there's lots of things going on, but how would you summarize your main research interests in your lab? The main kind of questions you're interested in answering, or the main things you're interested in?
So basically, as a postdoctoral researcher at the National Institute on Drug Abuse, we were very interested in how the brain actually represents information related to drug and the cues that are associated with that drug experience, so these so-called drug cue associations. When I came to Sussex, I realized, as much as it was interesting to study how the drug stores those kind of memories, the drug cue associations, retrieves them, somehow I think our brains really did evolve to store memories about things like food, which are very important for our survival. And I thought, well, why not study how the brain establishes and retrieves associations related to food and the cues that predict food availability. And somehow that became fascinated. And I think coming here, you're surrounded by nature, the Sussex Downs, and just saw a lot more wild animals, wild birds, badgers. And you do think wow, how did they survive in the wild? And I somehow got more fascinated with that. And even these little garden birds, squirrels, come to my office too. So it's the perfect environment for that. But we are interested in how these associations are established and retrieved in the brain. And we look at these specialized group of neurons called neuronal ensembles, which are just a few percentage of these neurons in the brain which are activated by stimuli such as food-associated cues. And we were wondering, which brain areas contain those ensembles. If they do, how are they regulated once we update these associations? So let's say normally this cue might predict food availability, but what happens when the cue no longer predicts that food availability? And that's called extinction learning, when we form those inhibitory associations and suppress those responses to those cues. So that's what we're interested in. We look at the nucleus accumbens, which is a brain area implicated in reward, things like triggering motivation, and so on. Yeah.
Mm-hmm. So, in a sense, you ended up kind of going back to sort of more basic, sort of the underlying process. So you started with drugs of abuse,
yeah,
and then ended up looking at, well, if the way those associations form is very similar to food, why not look at food cues directly?
Yeah, that's really going back to the basics, and I find it equally interesting, and it still has a lot of relevance to things like drug addiction, because that's also drugs are also rewards. They activate the brain very differently from natural rewards, but you also form those kind of associations with those drugs and cues. And if you understand the basic principles behind that, I think we could better understand conditions like addiction, in which these learned associations are involved. Yeah.
So, if you wouldn't mind, could you give us an overview of kind of what's the most, if you had to pick the most exciting project going on in your lab at the moment related to that, those interests? What would you say?
Yeah, so I'd say, well, I'll just talk about the current stuff that was going on that I'm quite excited about. So one of them is looking at a phenomenon called cue-potentiated feeding. So as the name suggests, it's when food-associated cues basically trigger you to eat more than you really need to, even when you're sated. Now, this has been shown in humans. Martin Yeomans, together with colleague Hans Kronberg, has done this with students, that food-associated cues will make students eat more. You could demonstrate that in rats as well as mice. And our current project is basically, how does the nucleus accumbens act, or has become activated when a mouse is in a food-associated context, and it starts to lick way more sucrose than when it's in a neutral context, not associated with food. So I think what does the activity look like? And we use a tool called fiber photometry. It allows viewing a real-time live broadcast of brain activity, so we could see what happens when the animal approaches this sucrose delivery site, as well as what happens right after it licks the sucrose, and then try to make sense of what activity patterns drive this behavior. And it might explain why we tend to overeat in the presence of these food cues.
I was going to say it's always interesting, or it's always nice when people choose a phenomenon that we can all relate to. I think you mentioned things like that. I think everybody's been in that experience where Christmas springs to mind, or I suppose Thanksgiving for Americans, is that all those associations are formed with those sort of settings where there's always lots of food and you always have more than you need to. It's that kind of thing.
Yeah, and those cues definitely invigorate it when, I don't know, you see other people eating it looking happy, or you smell the turkey, or you see the chocolate cake. And it does promote that behavior more and more. And it's quite important in this society now where you have a lot of fast food establishments, for instance, with very distinct cues, like the McDonald's Golden Arches. Is that a copyright infringement if I say that? No. But they obviously have very distinct contexts inside of McDonald's or in Burger King. And I always wondered, does that actually promote you to eat more once you're there, or you see their advertisements?
Definitely.
So that's one of the projects, and the other project is about how something called environmental enrichment, which is basically giving animals a much larger cage, more cognitive and physical stimulation, like giving toys, more tunnels, more room, they have to look for their own food. Basically how that kind of enhanced cognitive and physical stimulation, just briefly for a day, can reduce their reactivity to food-associated cues. So we've shown that even a day of that can reduce the amount of sucrose they seek out when they're presented with these cues. That normally motivates them to look for more sucrose. We recently found that that same enrichment reduces the amount of sucrose consumption. So they drink less sucrose, and it's interesting because in humans, even brief physical activity like 15 minutes on a treadmill, or even cognitive activities like 10-15 minutes of Tetris can reduce food cravings.
Oh, really?
Yeah.
Okay.
So even the NHS would say if you want to reduce cravings, I don't know, do something active. Go read a book, go walk, go listen to music. So there's something about that stimulation which reduces the drive of those cravings, maybe as well as how you process stimuli related to food. So our postdoc Kate Peters, her role is to try to find out in this area called the medial prefrontal cortex, which is involved in motivation, how's the activity of that area changing once an animal experiences that enriched environment and it's presented with a cue.
I suppose that has implications for, like you say, with the NHS, that has implications for kind of public health initiatives and things like that, in that if people are, struggling, have sort of obsessive or people are addicted, let's say, or have cravings for, lots of different things, I suppose there's a germ of an idea there of potential ways to help alleviate those cravings in terms of activity or things like that. It might help form some underlying basis for treatments.
I think so. I mean, I think a sedentary life, for example, is probably what is behind some of the let's say, excessive food consumption, for instance. And I think just having more of those moments where you can kind of engage in different activities, I don't know, somehow being enriched might certainly help reduce some cravings and perhaps somehow even reduce stress in some people. So definitely there's a lot of health implications for this. Yeah.
I'm always interested when I speak to people who do research in life sciences, because often there's a drive for people to try and find cures for things.
Yes.
People like studying disease because they like the idea of they're going to cure X, Y, Z.
Yeah.
And I'm interested whenever I speak to people that study or study things relating to psychiatric disease, things like depression or addiction in this case, because it's always an interesting question. Would you say you're more motivated in an attempt to try and cure, in air quotes, sort of addiction or addictive behaviours, or are you more interested in the studying of it to potentially lead to things like that, or is your drive more, I want to try and understand it so I can help treat people?
Yeah, I mean, definitely I do realize the wider picture that there are some public health issues. However, I think in my lifetime, at most I can try to reveal or generate fundamental knowledge that helps us better solve these problems. Our former department head at the National Institute of Drug Abuse, Roy Wise, he learned about drugs, brain, and behavior. He was doing a lot of dopamine research in the 70s. He told us young researchers, at most in your scientific career, your work will become one line in a textbook. As in, it's a very conservative process until many people replicate what you do and say, all right, 10 labs see the same thing. I think we can write this in a textbook that this is a robust, predictable phenomenon. So being realistic like that, I don't think I could, it's not that you can, within 5 years, you say, I do this research and there's a wonder drug, everybody's cured of their food cravings and everybody's happy. Because, I think progress in medicine like that, especially related to psychiatry, to the brain conditions, where you have a very complicated circuit that's more complicated than computer, will take more time to really resolve. I think that it won't be just a single pill that you take and your condition is treated.
Yeah. I agree, and I think people can end up into trouble when they sort of focus on, or when people start to think that they should be interested in. What's the phrase that?
Yeah.
When people are interested in something purely as an attempt to try and because they want to cure it or fix it. Or whatever, I think that especially in neuroscience that can lead you into trouble, because the chance of curing things like this that are so complicated is a difficult thing to define, especially with psychiatric disease, where there are already cures in psychotherapy and things like that. I'm always interested to speak to people who study these things, because it's interesting to hear from a scientific perspective, from someone active in the field, what they think about the kind of balance there between trying to alleviate people's suffering, but also recognizing that is a difficult thing, and there are other ways to do that, not through medicine, sort of thing.
Yeah, I mean, one has to be realistic of how slow the development of these things is, until from so-called bench to bedside approach of drug discovery to really delivering these drugs. I once applied for a job at a pharmaceutical company, Solvay, which is now, I think, defunct. One person I talked to said, "Don't get your hopes up too high. I've been working in this company for 30 years, and only one drug has made it to the market."
Wow.
So just be realistic, so enjoy the discovery process, that you are revealing something interesting about a complex system, and that humanity somehow learns from that, and we benefit, and perhaps as a result, that can lead to something much better. And I guess it is true for things like, just the initial studies on action potential. It's just purely fundamental work, but it helped us understand how we sense the world. This is how information is transmitted along an axon, that it's electricity. If you think about it, there's more recent studies showing that you can alter electrical activity in the spine, for example, to help people restore movements. But I don't think Hodgkin and Huxley were thinking about that probably at that time. But I don't know, slowly but surely knowledge builds up, and now we can actually create something better. And we see that for the mRNA vaccines as well, the initial discoveries of the mRNA, delivering mRNA to cells and to the vaccine. So it's a slow process, but
that shouldn't be discouraging to people.
No, definitely not. I think it's quite a noble thing to contribute to science, to better understand the world, to generate curiosity in the world.
And it's I can't remember the full analogy, but it's a bit like you're planting seeds that you'll never see fully grow to their full height, but that one day someone will do, and so it's kind of like it's quite noble in that sense that you're leaving something for the future, even if you won't fully see the full you can't predict the full extent of what you've sort of set in motion, I suppose.
Yeah, that's definitely true. You have to just do it, because generation after generation things might get better.